All right. Another Friday. Have your exams graded. I'll hand those back to you when we're done with what we have to cover today. Do you guys have any questions over what we've been talking about? You guys never have questions. Why is that? Yes, sir. They are in my office. Yeah. Yep. Okay. Why? I'm gonna have to reboot my computer. Is football team away today? Okay. That explains why some people are not here. Right. So we've been talking about genetics in kind of general terms in terms of inheritance. So there are a variety of different ways of thinking about genetics. One way is to think about genetics at a molecular level. So for this is what we talk about when we talk about kind of the structure of DNA, the order of nitrogenous bases in DNA, how that gets transcribed into messenger RNA, how that messenger RNA is then used to build a protein. And it's also when we talk about mutations and things like this, how those changes in DNA lead to changes in protein sequences. Those protein sequences then determine what your phenotype is, what your outward expression of those genes are. Another way of thinking about genetics is in terms of inheritance. And so I took you through how to work through dihybrid crosses, monohybrid crosses, dihybrid crosses, looking at how genes get transmitted from one generation to the next generation. That involves knowing how meiosis works to a certain degree. So it requires relying on that information from the first exam. But then, because meiosis is the method by which gametes are produced, you need to know what those gametes are in order to combine those gametes in a genetic cross, in doing something like a monohybrid cross or a dihybrid cross. Then we took this and expanded this idea from just characteristics that are binary, like pea plants. You have either green seeds or yellow seeds. You have inflated pods or smooth pods. You have round seeds or wrinkled seeds. We expanded that from those kinds of things to these quantitative or additive genetic traits. These traits that are underlain by multiple genes. So things like milk production in cows, the number of kernels in a ear of corn, the amount of beef that you can pack onto the skeleton of a beef cow, etc. Those types of quantitative traits are determined by multiple genes. Each gene adds something to the phenotype. And that's a different way of thinking about genes. Even our biology majors in my first year bio class, we're in the process of starting to go through this type of stuff. With them, they've been exposed to traits like flower color, seed color, and stuff like that back in high school, but thinking about genetics in this way is new to them. And so this is something that you more than likely have not seen because they haven't seen this before. But it's relevant because this is how most of the traits that are of interest to farmers, they would be determined by these types of genetics. And then we can think about genetics at the molecular level. We can think about genetics in terms of inheritance from one generation to the next generation. But then we can think about genetics in terms of things like genetic modification and genetic engineering. Two ways of modifying genes are through modifying organisms are through artificial selection. We saw artificial selection in the corn oil example that we looked at in class, where if you select for more corn oil, the population responds by accruing more of those dominant alleles that are responsible for oil production. And you see that the population of corn responds by having more oil. If you select for less oil, it responds by having less oil over time, over many generations. But then we talked about the second way of modifying genes, which is the method of genetic engineering. So I think of genetic engineering as a special case of genetic modification. And that's what we were in the middle of when we ended last time. Does this help you structure the information at all about genetics? Is it not helpful in helping you structure that information? Okay. So when you think about getting ready for the next test, you need to think about knowing how this process works. How do we go from DNA to messenger RNA to a protein? What is involved in those steps? If you don't remember those steps or you have trouble understanding those steps, come by and see me. We'll go through it again. Work the crosses. You can review crosses that we went through in class. You can also go and work crosses that are in that worksheet that I posted on Moodle. The whole purpose of that, you'll notice that I'm not taking that up. I'm not grading that. The purpose of giving you homework like this isn't to boost your grade, because I'm not grading it. The purpose of homework like that is to help you learn the thing that I'm wanting you to learn. And so work the crosses. Remember that the first thing that you have to do is you have to figure out if you're given the phenotype of an organism, you have to infer the genotype. Or if you know the genotype of an organism, that should tell you what the phenotype is. Once you've got all that worked out, it's just a matter of making the gametes and then combining them to make new offspring. And so it's a pretty simple stepwise procedure. But once again, you have to go back and remember those things about mitosis and meiosis in particular that we did because of the way gametes are formed. This is a mathematical idea. The relationship, the resemblance between offspring and their parents. And so we determine how heritable a trait is by looking at the parent phenotype and looking at the offspring phenotype. And the slope of that line relating those two phenotypes to one another, that is the heritability. And we can use that heritability to then determine how well that trait will be transmitted to the next generation. And then if we know what kind of selection is happening on that population, we can project what will happen over evolutionary time. A very different way of thinking about inheritance than this way of thinking about inheritance, but they're both useful ways of thinking about inheritance. When we do artificial selection, when we select for egg production in chickens, we're acting on this kind of quantitative genetic variation. We decide how much selection we want to exert, and based on that degree of selection, we should be able to predict how quickly the number of eggs that chickens produce will increase or decrease, depending on whether we're selecting for increased production or decreased production. Now, there's no reason why you would select for decreased production as a farmer, but you would select for increased production. But you can work out how quickly you want your population of chickens to change. Well, based on the heritability of that trait, you can then say, well, I need to exert this amount of selection in order to get that trait to move in the direction that I want it to move at a particular rate. And then genetic engineering is this, not particularly new, but some of the methods that we use for genetic engineering are relatively new. The newest of these is CRISPR, the CRISPR-Cas9 system. So that's where we ended up last time. That's a brief review of kind of where we've been in the genetic section of the class. You guys have any questions about this now that I've reminded you stuff that we've been through? Okay, all right. So CRISPR is this relatively new method of altering the genome of an organism. It's a genetic system that was borrowed from bacteria. Remember that when we get infected with a foreign agent, either a bacterium or a virus, we have blood cells that react to the substances that are on the outside of that virus or the outside of that bacterium. And then our blood cells create antibodies. The antibodies, the next time you are exposed to that thing, the antibodies will bond onto the outside of those viruses or bacteria. And it will, in some cases, just isolate them. In some cases, it will actually break them open and destroy them, depending on the type of interaction that the antibody has with the organism. Bacteria don't have a blood system, so they don't have this ability to create antibodies to antigens of foreign invaders. And so they have evolved a different way of dealing with viruses, which basically bacteria are only affected by viruses. Bacteria don't generally get affected by other bacteria. And so when a virus invades a bacterium, it basically injects its genome into the bacterium because the bacterium doesn't have a nucleus like we have. We have a nucleus, plant cells have a nucleus where our genetic material is kind of protected from the rest of the other parts of the cell. Bacteria don't have that. Their chromosome is just free-floating in the cytoplasm of the cell. And so it's very easy for the viral genome to then just insert itself into the genome of the bacterium, the same way a scientist would insert a gene into the genome of the bacterium, if we're using the old style of genetic engineering. And so what bacteria evolved was a system by which they can recognize DNA sequences that aren't their own. They can recognize DNA sequences that correspond to the DNA sequences of viruses. And when they find those DNA sequences, they can just go in, cut that viral DNA out of its genome that's been inserted into its genome by the virus, cut it out and get rid of it and dismantle it. And that's how they prevent being infected by viruses. So a couple of researchers who were studying how bacteria fend off diseases themselves discovered this system. And it's called the CRISPR-Cas9 system, and it's made up of two parts. One is what we call this guide RNA molecule. And the second part is a Cas9 protein, which is a protein that has enzymatic function. It's the thing that actually does the cutting of the DNA. So the guide RNA is called the guide RNA because this long strand of RNA sequences can complementary base pair to sequences in the bacteria's genome. And it will match those DNA sequences of viral DNA. And so the bacterium produces a number of these different target-specific RNA molecules that will match up to the DNA sequences of the types of viruses that it has come into contact with. And so through complementary base pairing, the same way that DNA bonds together with the other strand of DNA, this recognition protein, this guide RNA, basically will bond to the DNA sequence of the virus. Once it does that, attached to this is a protein called Cas9, and it has a variety of different functions. In some cases, it will just cut DNA. In other cases, it will insert DNA. And in other cases, it will cut DNA and then put that cut strand of DNA back together again so that it essentially deletes sections. If you have a DNA molecule, give me a minute. If you have a DNA molecule and Cas9 comes in and cuts it in a particular spot, now rather than one long DNA molecule, you have two shorter DNA molecules. And if there was a gene that extended from here to here in this DNA molecule, that gene no longer functions because you've disrupted it. You've broken that gene now into two separate sections. And while transcription may start here, and you might transcribe a messenger RNA molecule, that messenger RNA molecule will be shorter, which means that the protein will be shorter, and as a result, it won't be functional because it's missing the other half of that protein molecule. And so the act of cutting DNA just basically results in a gene that loses its function. It used to work, and now it doesn't work anymore. That's kind of the easiest, sloppiest way of getting rid of foreign viral DNA. Just cut it in the middle, and then that viral DNA doesn't work any longer. You can insert a gene, which means cutting the DNA open. Inserting the gene of interest. And then putting the DNA molecule back together again. The guide RNA is made up of two different sections, a target-specific sequence, but then there's this other section, and it's this other section that is going to be used for adding genes to a genome, and we'll see how that works in a few minutes. But then the last thing that it can do, it can cut and put the DNA back together again. This basically just deletes unwanted DNA. And so you see the Cas9 protein sometimes represented in talks that are given to the general public as kind of a Swiss Army knife. So it's a type of protein that has a lot of different enzymatic functions such that it can kind of do whatever is kind of needed at the moment. And so you see this Swiss Army knife sort of image here. But basically, it's just an enzyme that has the ability to do all of these different things. So the guide RNA combines with the Cas9 protein into this complex of a protein and a messenger RNA. The messenger RNA guides, that's why it's called the guide RNA, guides the Cas9 to where the Cas9 is supposed to work. And in this case, it can do a variety of different things. It can, in this case, it's actually inserting itself, so it's through complementary base pairing, the target specific sequence is identifying the section of the DNA molecule where it's going to bring the Cas9 protein. And in this case, it's just cutting that DNA and doing nothing more to it. In some cases, bonded to the Cas9 protein, you might have a genetic code that represses or downregulates a gene. So remember that during protein synthesis, the way you get protein synthesis to occur is you basically turn on a gene. You tell that gene to start the process of transcription of that gene from DNA into messenger RNA. Well, genes get upregulated and downregulated throughout time inside of your cells. In some cases, the genes are really active in transcribing copies of themselves. In other cases, they're not very active. This is almost always determined by the presence of some other chemical in the cell. So, for example, if you need a particular protein that is involved in nervous transmission, that will generally respond to some stimulus that signals to the nerve cell that it needs to send transmissions. And so we have different types of compounds that serve as repressors that depress gene transcription and activators that enhance gene activity. And so you can basically turn a gene down or on, or turn it down or turn it off altogether just by having attached to that Cas9 protein a compound that will do that. Or you can turn the gene on or turn it up by having another type of compound. And so this Cas9 can not only open up the DNA, but it can also then have an effect on a target gene by either tamping down its expression or by ramping up its expression so that it makes more messenger RNA. And so this system can not only just alter the DNA, in this case, they're not altering the DNA at all, they're actually just adding things that interact with the DNA, bringing them to the right gene so that you can regulate that gene precisely. By either knocking the function of that gene out or by ramping up the production of that gene. And then the third thing, which they don't have a graphic of, is you can also cut the genome here, have genes that are attached to the other part of this messenger RNA, and insert those genes in and actually alter the sequence. In some cases, you can do that with long stretches of DNA. In some cases, you actually just want to have it cut out one nucleotide and put a new nucleotide in place of that. And that's exactly what they did with this woman who has sickle cell disease. The mutation that calls sickle cell disease is a single nucleotide change. So there's just one nucleotide that is different that then changes the structure of the protein such that it's not normal hemoglobin, it's this altered hemoglobin. And CRISPR can very easily identify where in the genome that single nucleotide is cut the old nucleotide out, put the new nucleotide in. It's like changing a flat tire on your car, and then you're on the road to healthy blood cells after that. So this has been used a lot in experimental model organisms. So we've been able to alter, I don't do this because I'm an ecologist, but scientists who do this kind of thing have been able to alter gene expression in nematode worms, fruit flies. I think this is a frog embryo. And they've also been able to do this in plants as well. And so it's this general kind of all-purpose tool that people are finding that they're able to use to alter the genomes of lots of different kinds of organisms. And the most recent thing that has happened is this woman that has agreed to have her blood cells CRISPRed. Question about the technique. All right. You guys went and listened to the NPR programs, hopefully. Did you? Okay, good. The first thing I kind of want to know from you is, what are your initial impressions after listening to those? I realize they were both about 20 minutes or so apiece. One of the things that I like about NPR is they tend to devote a sufficient amount of time to a story to really delve into the story in ways that you won't get on network TV and things like that. So what are your initial impressions after listening to those two articles? Yeah.
Seems pretty impressive. Again, the first one she mentioned that they said something that probably won't fix all of them, but the second one, she said that she seemed to was completely sure of it.
Yeah, so it's a pretty radical sort of procedure. Remind me once again, what did they have to do to accomplish this in her?
I think they had to inject her with stem cells.
Okay, so the first thing they did was they took stem cells out of her. Then those are the stem cells that they altered using CRISPR. Then what did they have to do?
They injected it back through.
They injected it back into her, but what did they have to do before they did that? Because if you just take the small number of cells that have been CRISPRed and put them back into her body where all these other stem cells are that have the sickle cell mutation, they're going to totally overpower whatever the scientists put back into her. So what did they have to do? She went through chemo, and the chemo was focused on killing off the stem cells that create her red blood cells. So they basically stockpiled a bunch of her blood that was sickle cell blood, so they give her transfusions. Then they killed all of her stem cells that would normally make red blood cells. They then had to give her transfusions in order for her to replace the red blood cells that she could no longer produce. And then once they got her stem cells CRISPRed, they put those stem cells back in there, sustained her on her own stockpiled blood until those stem cells started producing the blood that was not sickle cell blood. And then eventually the sickle cell blood cells break down because blood cells just don't stick around in the body forever. They break down over time. So it wasn't a casual procedure. Like this person didn't wake up one day and say, "Hey, I think I'm going to go have myself CRISPRed and I'll be all better." It was a long, drawn out, involved process. Involved chemo. How many of you guys know somebody who's been on chemo? Yeah, what are your impressions of chemo from talking to them or seeing them? You don't want it, do you? No, my mom was diagnosed with breast cancer a number of years ago. She went through chemo. She lost all of her hair. She was crabby for like 9 months, crabbier than normal for 9 months. Um, in the end, she's fine, but still, chemo is designed to kill your cells. Like that's what chemo does. And so the trick with cancer, treating cancer, is when you're given chemotherapy, The goal is to kill your cells enough that it gets rid of the cancer while avoiding the line that you cross over where you've killed your other cells so much that you end up dying. And so chemo is not a happy time. What about the rest of you? Similar impressions of what chemo is like? Yeah, you want to try and avoid it if you can at all avoid it. So it wasn't a uncomplicated, seamless, easy sort of procedure. Uh, it seems like in the end, she seems like they've actually corrected a genetic condition. What are your other thoughts on this after you have listened to the stories? Yeah,
I thought it was interesting that they actually talked about her, like excitement to go back to her family and how this was going to be like affecting her kids and her family life.
yeah. When we think of healthcare, a lot of times we just think of healthcare as being, getting the fix for whatever ails us. Um, over this, well, okay, so recently, recently I got bit by a cat. Still have the scabs on my leg. I was working in a barn. There's this old cat. Her name is Skitsy. Lives at my wife's family's farm. Hangs out in the barn. I was in the barn working. This cat is so old that she's lost the ability to meow. But nobody ever comes out to the barn. I've been working on a project out in the barn for a couple of months now, and so I'm like the only dude who comes out to the barn. And so she's like, "Oh, a human coming out and spending time with me in the barn." And I'm like, "I'm not spending time with you. I'm doing something else, and we just happen to both be in the barn." So she comes up and rubs on my legs and stuff, but she doesn't make any noise. She's old and very frail and thin, and so she doesn't make any noise. She doesn't meow. She doesn't do anything. And one day I was putting together a cabinet, and she curled up right behind my legs. And I stepped back to tighten down a screw on this cabinet, and I stepped on her tail. But because she's lost her voice, she didn't meow or anything. She just bit right into my leg. And if you have ever been bitten by a cat, cats have amazingly septic mouths. And so about 2 weeks ago, um, I guess was when this happened. And after a couple of days, you could see that my leg was getting redder and redder up and down my leg. It was basically turning septic. So normally when you think of healthcare, you think, oh, I got bit by a cat. I'm getting septicemia. I need to go see the doctor, and the doctor will fix it. That's I think oftentimes how we think about healthcare. Now, you can die from septicemia, and so I was glad that I went to the doctor when I did. Luckily, I know the signs of septicemia. Um, he gave me a double dose antibiotic, and it knocked the infection out, and I'm successfully on the road to healing, although you probably don't want to look at my scabs any longer. Um, but we don't think about oftentimes medicine and medical treatment and healthcare as something that can affect your quality of life. In this case, this woman had a really pretty severely degraded quality of life because of this genetic condition. And also, it's a quality of life that is eventually going to kill her much younger than she would have if she hadn't done this. And so, I don't know if you guys went online and did any research about sickle cell disease, but you die younger, much younger, if you have this condition, especially if you have the most severe forms. And before you die, you spend a lot of your time in the emergency room. Um, People with sickle cell have many more emergency room visits over the course of their lifetime than regular people do as a result of this chronic condition. So for her, yeah, it is really a quality of life issue as well as a longevity of life issue. So yeah, that's a good observation. What else did you think about this as you were listening? I don't know. Um, I haven't heard of any, but I haven't really dug very hard looking. That's a good question to have. Um, One of the tricks about doing these types of kind of radical experiments on humans is that they're kind of frowned upon. In the same way that people were concerned about the quick emergency use authorization of the COVID vaccines. One of the concerns, and this is a very realistic sort of concern to have, is, do you really have enough data to say that it is both safe and effective? And that's a legitimate question to ask about those vaccines. In the same way that there is the legitimate concern about this treatment. Okay, so this treatment fixed the sickle cell. Well, are there other consequences of having done that? Hopefully, the thing about CRISPR is that it's so specific to where it acts within the genome that in theory there shouldn't be any additional complications of that, because that guide RNA takes the CRISPR Cas9 complex to the exact place in the genome that it needs to be to do its work. This part should keep it from having broad scale effects, but at the same time, you never know until you do it. One of the other difficulties with this is that they now have a sample size of 1. And one of the things that happened with the COVID-19 vaccine is that they had to wait until they had enough data. So they got emergency use authorization from the FDA, and then they gave it to a lot of people. They followed a lot of those people. They've now given it to millions of people. They've now followed tens of thousands of those people to look at outcomes, to the point that now they have enough data that they applied for the full approval of the FDA, and they got it for the three, well, 2 of the 3 vaccines that are currently being used. But at the beginning, they didn't have enough data for the FDA to say, yeah, full approval, go for it. They said, things are looking good, we'll give you emergency approval, but one of the things that you have to do is you have to continue gathering data on the people who are getting the vaccine to put into your application for full approval. So we now have a sample size of 1. And as a statistician, I have a graduate minor in applied statistics. A sample size of 1 is just bogus bullshit. You can't say anything about the general applicability of this based on one person's experience. Now, one person's experience turns out seems like it was pretty good, but you have to get approval to now try this in a bunch of other people. And you have to go through university committees. We have one here, it's the Institutional Review Board. Anytime you do anything having to do with students, with humans, you have to get your study approved by the Institutional Review Board, even if it's just taking a survey of students. And so you have to pass a number of hoops, and the scrutiny that you get ramps up with the unusualness of the thing that you're proposing to do. So if you're proposing to now find 10,000 sickle cell disease patients in the United States and CRISPR all of them, that's going to require a great deal more scrutiny than trying it on one individual who, quite frankly, is a volunteer. And so, yeah, I'm not sure if they've done it on anyone else. It seems like this is one of the most obvious applications of this, because sickle cell disease isn't a disease that you catch, it's a condition that you inherit. And that's a classic kind of thing that you would want to use something like CRISPR on. And so it wouldn't surprise me if the people who did this are in the works of trying to get approval to do a larger study. I just haven't heard about it yet. Yeah.
So like with the cells they took out and put back in, be specific to her, or could they like use those cells?
Yeah, so this is a tricky thing about the human immune system or animal immunity at all in general, is that In the same way that there are substances on the outsides of bacteria and viruses that signal, I'm a bacterium or I'm a virus, and our immune system responds to those, you also have those things on your cells. So you have cell markers on your cells that are unique to you. And this is why we can't just give anybody else's blood. If you look at blood cells, I don't know if you guys talked about blood typing at all in your high school biology class, but in your blood cells, we have type A blood, type B blood, type AB blood, and type O blood. And if you look at a blood cell, there are glycoproteins, which is just a sugar and a protein smushed together in the same molecule, a molecule made up of a sugar component and a protein component. And we have antigens on our blood cells that can be the A antigen. We have cell markers on our cells that can be the B antigen. And what happens is if you have A blood, you have the A antigen. If you have B blood, you have the B antigen. If you have AB blood, you have both the AB antigen. And if you have O blood, you have no antigen. So this is the antigen that you have. But what that means is that when you make antibodies, you don't make antibodies to the A antigen if you have type A blood, because you would be making antibodies that destroy your own blood. So in terms of the antibodies that you make, A people make antibodies, B people make antibodies to A blood, AB people make no antibodies to blood, and O people make both anti-B and anti-A. So you have your own markers that signal to your body that you are you, that your cells are yours. And so if we take those stem cells, that they're not blood cells, they're stem cells that make blood, you also have, in addition to these kinds of antigens, you have your individual cellular markers that are on those, such that you can't just take this woman's blood now and her stem cells, give them to someone else, and then have it work in there, because chances are that person will reject those stem cells. In the same way that you can't just get somebody else's heart and transplant it into another person, you actually have to have a donor match. And when they're talking about that donor match, the question that they have is, are the cell markers on the outside of the cells of the donor person close enough to the cell markers of the recipient that the body won't then attack that new heart, because the body senses that it's actually new, actually not the person's. And even at that, when you get an organ transplant, you go on what they call immunosuppressant drugs, which suppress the immune system from functioning as well, to once again prevent the body from rejecting that transplanted organ. And oftentimes you're on those immunosuppressant drugs for the rest of your life. So once again, since COVID is such a large part of our lives, one of the worries with COVID-19 circulating in the community is that there are some people who can't take the vaccine. If you're an organ transplant patient, they could give you the vaccine, but your immune system is suppressed so that you don't reject your kidney or your heart or your lung or whatever has been transplanted into you. And so even if they gave you the vaccine, your body wouldn't mount an immune response, because the same immune response that would produce antibodies to COVID virus would also be producing antibodies to the organ that is in your body that is not your own. And so those people can't receive the vaccine. As a result, they had to be very careful about who they interacted with during the pandemic, because They can't just go hanging out with anybody because it's a real risk for them to get sick because they don't have natural immunity because their immune system is suppressed. They don't get the vaccine because their immune system is depressed. And so they're at real risk of getting sick and dying from it unless they're very careful about who they come into contact with. And so not surprising, a bunch of organ transplant people, they just became shut-ins during the pandemic because it just wasn't safe to interact with other humans. This is also why they blood type you. So it used to be thought that because O blood had no antigens, you could just dump O blood into A, B, B, or A people. It's called the universal donor is O blood. And this was fine until they got into wartime situations. This was mainly the first war where we used blood transfusions a lot as a treatment for wounded was in Korea. And during Korea, you had on your dog tags what blood type you were. And we finally knew enough about blood typing that we could do things like transfusions. And they would determine who could get whose blood based on this antigen system. And we used to think that you could just give O blood to anybody because this person produces anti-B, this person produces anti-A, this produces neither, but these two people won't react with O blood because O blood doesn't have any antigens on the outside of the blood cells. It basically looks like this. So there's nothing for these antibodies to latch onto. But what they discovered in Korea is that when you've had your leg blown off by a mine, you lose a lot of blood. And when you dump a lot of blood into a new person, you don't have to worry about that person's antibodies interacting with O blood. But if you put a large enough volume of O blood into a person, you have to worry about its antibodies attacking the blood of the recipient. And so we don't do the whole universal donor, universal recipient. If you go to the hospital, if you have a planned procedure, oftentimes they'll ask you to stockpile your blood beforehand so that they not only give you your blood type, they give you back actually your blood. And so in this way, you're kind of like the woman in the story. But if you're in a car accident or something like that, they don't give non-type blood to people. They give you, if you're type A, they give you type A blood. If you're type AB, they give you type AB. If you're type O, they give you type O. Because if you need a large enough transfusion, it causes problems because the blood that you're receiving may have enough antibodies in it that it causes problems. One of the things that has happened as a result of the COVID pandemic is blood banks are low on blood, chronically low on blood. So how many of you have ever given blood before? Okay, you should go give blood again. They need you right now because during the height of COVID, blood banks didn't have people coming to the blood banks to give blood because everybody was afraid of getting COVID. And so blood stores globally and in the United States as well are chronically low right now. And that's bad because we tend to not just give you any old blood. We want to give you the blood type that matches your blood type. So yeah, we can't take this person's stem cells and put them in somebody else for this cell recognition reason. The body that you put those stem cells into will recognize that those aren't your stem cells and they'll just kill them. Your body will just kill them. Other thoughts as a result of watching or listening to these stories? Yeah. Oh yeah, you had your hand up earlier.
And wouldn't this stop this particular genetic disease because they're altering her DNA? If she were to go and have children after this procedure, they wouldn't carry the DNA out of themselves.
That's the tricky thing. So the cells that produce gametes are not the same cells that produce blood cells. So they've altered one particular type of stem cell. They haven't altered all of the cells of the body. They've just altered those stem cells that produce blood. The cells that produce eggs and sperm are located in the ovaries and the testes. And so if you're going to do that, you would have to then find a way of altering those. And that's a little trickier because now you are wanting CRISPR to go into a fully formed organ and change all of the genetic code in all of those cells. And that becomes a much more complicated thing. So no, she would continue to pass this on to her offspring. If she mated with a homozygote, her children would be normal. They would have normal blood, and they would have the protection that you get from being a heterozygote. But if she happened to mate with somebody who was heterozygous, half of her offspring would have the same condition that she has, because it didn't alter anything about any of the other cells in her body, just this one particular type of stem cell. But that's also a good question. In order to have this work for the entire body, you would have to do kind of what Agrobacterium tumefaciens does. If you could genetically test an ovum, so let's say that you know that you have this condition, you want to have offspring, they could take the eggs that you produce, and we can take eggs out of females all the time. This is how in vitro fertilization works. We could take the eggs that she produces, CRISPR those, combine them with the sperm of whoever she wanted to have the baby with, and then put those back into her. And because you've altered the egg, then any cell that comes from that egg through the process of mitosis would be genetically identical to the egg itself after it gets fertilized. And so in that way, she could have offspring that were free of the condition, but she wouldn't just be free to freely reproduce. She would have to once again go through an arduous process of harvesting her eggs, crisping her eggs, fertilizing the eggs, probably outside of her body, and then implanting those eggs into her. So she would basically be doing in vitro fertilization with a twist, with a CRISPR twist. Once again, a good question. Yeah.
Do you think like this method of like stem cell therapy or whatever you want to call it is going to start becoming more common?
Probably not immediately. I would assume it's expensive. Yeah. It's cheap enough to do that people do it with fruit flies. My wife is a geneticist at KU, and she has a project where she's CRISPRing fruit flies to knock out the function of some genes involved in copper toxicity. She studies how copper is processed by organisms, and she uses fruit flies. And so it's not so exorbitantly expensive to just do CRISPR. To do CRISPR on a human in this particular way, where you have to go through chemotherapy and stuff like that, I'm sure that was pretty expensive. Do you guys have any concerns about this type of technology? You guys seem all kind of positive, but only 4 people have really spoken. What are your concerns? What are your misgivings, if there are any?
I mean, I'm sure a lot of people would just consider it like playing God to a certain extent.
Sure. Have you guys ever seen the movie Gattaca? If people when this first came up, people were like, "Oh, it's Gattaca." Gattaca is this dystopian future science fiction film where they basically sequence your entire genome when you're born, and then they put you in a category, and you carry around that categorization for the rest of your life. It determines whether or not you go to college or not. It determines what kind of jobs they'll let you have, et cetera, et cetera. And this guy decides that he's going to buck the system. He has inferior DNA, and he pays a guy who has superior DNA, but is a quadriplegic from an accident. It was an accident, wasn't it?
It's been a long time.
Yeah, it has for me too. Pays a guy to basically harvest his cells to scatter around his apartment, scatter at his workplace, on his keyboard, et cetera, so that when they come by and genetically test, it doesn't look like Ethan Hawke's DNA, it looks like Jude Law's DNA. Because Jude Law is in a wheelchair but has superior DNA, Ethan Hawke is up and around and functional, and he ultimately wants to go off-world. He wants to go off of Earth, and the only way to go off of Earth is if you're one of these genetically superior people, because he just wants to escape all the bullshit genetic superiority crap. And so, yeah, people have that concern that you're just being way too godlike in your control of the genetic system. And other people are worried about, because in Gattaca at the beginning of the movie, they show Ethan Hawke's parents at the genetic counselor saying, "Oh, you have a child that's going to have these characteristics, but for a fee, we can upgrade your offspring so that he is one of now the genetically superior ones. But with each upgrade, it costs money, and they don't have enough money; they can't afford it, and so Ethan Hawke just becomes Ethan Hawke, and he lives in Seth Meyers' attic crawl space, and so there's that concern that this leads to designer babies and things like this. Um, One of the cool things about government, and this is also one of the bad things about government, is that they can place regulations on things. So sometimes you like the regulation that the government puts out, sometimes you don't. But one of the things that would have to happen if CRISPR became widely accepted is, or widely used is, under what conditions are you allowed to do this and on what conditions are you not? And that would have to be pretty heavily regulated. And because all of this stuff has to be done with trained scientists in facilities and stuff like that, the chances of bad behavior are reduced, if not eliminated. There's always the possibility for bad actors. The trick is ferreting those bad actors out and putting them away. Another function of government. Other comments or concerns? This is new. It's cutting edge. We don't know what a lot of the answers are for what we could use this for in the future and what it means for kind of the genetic future of humans. But it's something to contemplate. It's something that you guys should certainly be aware of out in the world. So